In this lecture, we will be discussing the development of the vertebrate body plan. That is, we will take a trilaminar embryonic disc, and through a series of folds, we will develop an embryo in which now there is a ventral surface, a dorsal surface, a right and left surfaces, and a body cavity. As these folds occur, we will see the development of, first of all, the ectoderm. The ectoderm will give rise to the neural tube and the nervous system, as well as the surface ectoderm. We'll then see the development of the endoderm, which will give rise to the epithelium of the GI tract, the respiratory tree, and the pharynx. Finally, we'll discuss the development of the mesoderm and discuss how the mesoderm can be subdivided into paraaxial mesoderm, intermediate mesoderm, and lateral plate mesoderm. This is a review of a previous lecture in which we talked about the blastocysts having a trophoblast and an inner cell mass. The trophoblast gave rise to the cytotrophoblast, which gave rise to the syncytial trophoblast, and these are the components from the fetus that contribute to the placenta. We also talked about the inner cell mass and how that gave rise to the epiblast. The epiblast then gave rise to amnioblasts that line the amniotic cavity, as well as the embryonic epiblast, which formed through the primitive streak both the embryonic endoderm and embryonic mesoderm. And then finally, that embryonic epiblast became the embryonic ectoderm. So we will now talk about the embryonic mesoderm, embryonic ectoderm, and embryonic endoderm. Here is that trilaminar embryo. To review, we have an amniotic cavity, we have a yolk sac, the embryo sits within a chorionic cavity. It is a trilaminar disc at this point with an ectoderm in blue, mesoderm in red. The notochord sitting in the midline is purple, and then the yellow is the endoderm. The mesoderm itself can be subdivided into paraaxial mesoderm, intermediate mesoderm, and lateral plate mesoderm. To begin, we're going to talk about the ectoderm. We're going to look down on that developing embryo again. This is the epiblast that we saw in the previous lecture. This reminds us that there was a primitive node and a primitive streak, and that the cells were migrating through the primitive node and streak to form the mesoderm and the endoderm. In particular, we want to look in the region of the primitive node, as these cells of the epiblast are going to migrate in through the primitive node and then migrate in a cephalic direction. So as the primitive node forms, cells are going to migrate through the primitive node and then begin to move back and form the notochord, which we see here. Here we can identify the notochord, and the notochord is going to move in a cephalic direction. It will continue moving in a cephalic direction until it reaches a level where there will be some intermediate some mesoderm. This is called the prechordal plate, and then a region where ectoderm and endoderm meet without intervening mesoderm. When we think about the formation of the notochord and as its movements, we can think about what are some of the events that are taking place that lead the notochord to induce the overlying ectoderm to become nervous tissue. This slide talks about the fact that bone morphogenetic protein and fibroblast growth factors are important in forming the intermediate and lateral mesoderm, and that in the midline what we're going to have is the expression of cordon noggin, and follistatin. And what they're going to do is they're going to inactivate bone morphogenetic protein. And what is going to happen is there will be a gradient of BMP from very little in the midline to a significant amount as you move laterally. And this gradient that forms in bone morphogenetic protein is important in terms of the formation of the nervous system in the midline, as well as mesodermal structures out laterally. So cordon, noggin, follistatin are going to be important in allowing for the formation of the paraaxial mesoderm on either side of the notochord. And that paraaxial mesoderm is also going to be important, along with the notochord, of inducing the overlying ectoderm to become the neural plate, which is what we see here. So we have cordon, noggin, and follistatin up in the cephalic area where the head will develop, and fibroblast growth factor and WNT3A more caudally, that are going to influence the development of this neural plate. Intermediate between the neural plate, which you see here, and the lateral area of the embryo, we're going to have a gradient formed, and in this region here there's intermediate levels of bone morphogenetic protein. This then allows for the expression of PAX3, which then upregulates FOXD3, and this will lead to the formation of neural crest cells. So it is at this area here, the junction of the neural plate and the rest of the surface ectoderm, that an intermediate level of PAX3 is going to allow for the induction of a new group of cells called the neural crest cells. Neural crest cells are formed with the induction expression of FOXD3, as well as SLUG. SLUG is going to be important for allowing these neural crest cells to migrate, and we're going to be talking about that throughout all of embryology. So let's turn back then to this neural tube, and what we're going to see, this neural plate, and we're going to see this neural plate begin to round up on itself. In the midline, we're going to have a neural groove forming, and out laterally we're going to see some neural folds. Here you can see the neural groove forming. A little bit later, these neural folds, and by day 20 we can start to see some somites forming. This is somites that are formed from the paraaxial mesoderm. As the folding continues, the edges of the folds on either side are going to meet in the midline. They meet in the cervical area, or what would be the cervical area in the future, and that these folds are then going to fuse, and that fusion will move in a cephalic direction as the brain structures are going to form, and in a caudal direction as the spinal cord regions are going to form. And so a neural tube is going to develop. The fusion will continue cephalically and caudally, and before it finishes we have two openings, one in the cephalic area called the cranial neural pore, and in the caudal area called the caudal neural pore. And these will be the last areas that will fuse. If we look at an image from the University of New South Wales Embryology website, what we will see is the following. Here we see the embryo just prior to fusion. We can see the neural folds, the neural groove in the midline. We can see some somites, and here you can see a little bit later then the fusion of the neural folds into the neural tube, and this is occurring during the 4th week of development, from about day 22 to day 23. So if we look at this view of the embryo, looking down on the embryo, we see the neural folds have begun to fuse, and yet in the cephalic area we still see that anterior neural pore, and in the caudal area we see the caudal neural pore. As we mentioned, due to the gradient of bone morphogenetic protein, as you move laterally, there's an area signified in this cartoon by blue, and this will be the region where neural crest cells will form. And as the neural tube forms, just lateral to it we will have two masses of cells called the neural crest cells. Now, as the embryo develops, here we're looking at the embryo in sagittal section. Here we're looking at it. This is in sagittal section. If we make a section at this point, we would see the embryo in cross section. And what we want to do is follow the embryo in cross section, as lateral body folding is going to bring the endoderm together, the mesoderm together, and the ectoderm, the two edges of the ectoderm together. So we're going to talk about the ectoderm in brown, the mesoderm in green, the notochord you can see here in the midline, and the endoderm in yellow. This would be the yolk sac, and this would be the amniotic cavity. Here you can see the beginning of the neural fold that begins to form, and that neural fold, as it begins to form in the midline, is being induced by the paraxial mesoderm as well as by the notochord. Also notice out laterally that the lateral mesoderm is going to begin to split and a cavity is going to form. And here you can see this embryonic body cavity beginning to form out laterally, and you can see in the midline how the neural folds will continue to form and they will fuse in the midline. This forms the neural tube. The ectoderm is then going to lie over the neural tube, and not shown here, there would be neural crest cells. Notice out laterally we begin to get the folding of the ectoderm and the two layers of mesoderm. These layers of mesoderm then are going to continue folding around, and they will move towards the midline along with the endoderm and its mesoderm, and they will meet in the midline to fuse. So that at this point, at about 28 days, we have lateral body folding give rise to an embryo now completely surrounded by ectoderm. That ectoderm is lined by mesoderm, and this is called the somatic mesoderm. There is a body cavity that formed during this process as the two body cavities fused, and there is another layer of mesoderm that lines the endoderm, and this layer of mesoderm is called the splanchnic mesoderm. The endoderm then formed the gut tube, and as we move posteriorly, we see the notochord, we see some developing blood vessels, and we see the neural tube surrounded by mesoderm. This is a very nice animation of lateral body folding. And we can appreciate the complexity of these movements. And so if we look both at a cross section and sagittal section, we can look at lateral body folding. This is from the University of Indiana, and here we can see what happens during lateral body folding. Notice the embryonic spaces are going to move together, forming the body cavity. Notice the ectoderm is going to come around and fuse during lateral body folding. And notice, because of where we're looking here, in front of the umbilical cord, we're going to have the gut tube form. So now we have an embryo sitting within the amniotic cavity. Notice the amniotic cavity now completely surrounds the embryo. In addition, an intraembryonic body cavity then surrounds the gut tube. If we look at a sagittal section, we can see head and tail folding occur. So this will make the point that while lateral body folding occurs, so does head and tail folding. And as we look at this process, we can notice that the brain grows forward rapidly. As it does so, it will move the prospective heart tissues in a more caudal direction. It will form two outpocketings of the gut tube, which will be the foregut and the hindgut, and it will then leave the midgut in communication with the yolk sac. The yolk sac midgut communication is called the vitelline duct, and the vitelline duct, along with the umbilical vessels and a little bit of this structure called the allantois, are going to be the structures found in the umbilical cord. Notice again how the embryo is completely surrounded by the amniotic cavity. And so when we look at the embryo, at this stage, again going back to the University of New South Wales embryology page, what we can see is now an embryo at stage 11. Here you can see the somites, here you can see the fused neural tubes, except a little bit back here in the caudal neuropore. And when we look at this stage 11, what we're seeing is an embryo which is at about 28 days of development now, and the vertebrate body plan has been established. So that what we have is an amniotic cavity. We have within the amniotic cavity a neural tube, another tube, the gut tube, and then the body cavity. The embryo itself is surrounded by ectoderm. The gut tube is surrounded by endoderm, and in between we have mesoderm. When we talk about the ectoderm, then the ectoderm will form three sets of structures, both the surface ectoderm structures, things like the epidermis, hair, nails, etc., including things like the anterior pituitary gland and enamel of the teeth. The neural crest cells will form things like sensory and cranial ganglia, cells of the adrenal medulla, and other structures. And the neural tube cells are going to form the cells of the central nervous system, the retina, the pineal, and the posterior pituitary. And this is the part of the course which we'll be starting at during every lecture. So we'll be talking a lot more about this as we move on. If we look at the endoderm now, we can talk about what happens to the endoderm as we get the formation of the pharynx, the GI tract, and the respiratory system. And we not only need to consider lateral body folding, but as you saw previously, head and tail folding. And the two together will leave the endoderm in three segments. One segment is the foregut, another the midgut, and the third the hindgut. Here we're looking at the embryo and sagittal section. That is, if we look down on the embryo from this view and make a plane a section sagittally, this is what we would see. Again, at this point, that's the chorionic cavity, that's the amniotic cavity, and this is the yolk sac, and there is the trilaminar disc. And what's going to happen is during head and tail folding, you're going to get rapid elongation of the neural tube, and as that neural tube moves forward, it will continue to push the heart region in a more caudal direction. In addition, what it's going to do is leave the gut tube in two little cul-de-sacs, one anteriorly here, cephalically, and one caudally. And what you're going to end up with then is a foregut, a midgut, and a hindgut. Notice that the foregut is in contact with the ectoderm at the region of the oral plate. The oral plate is that region where the prospective oral cavity is going to meet the prospective pharynx. And so the oral plate would be in about the region of the anterior tonsillar fossa. If we go from the foregut to the midgut, noticing the midgut is in connection still with the yolk sac, via the vitelline duct. And then as we move backwards, we can identify the hindgut, which is going to elongate or enlarge here caudally as it will receive communication from the atlantois. Together they will form a large structure called the cloaca. The cloaca will come in contact with the ectoderm in the region of the cloacal plate. And that's where endoderm and ectoderm meet, and that is the region of the anal canal where the upper part of the anal canal is derived from the hindgut, and the lower part of the anal canal is derived from the ectoderm. So what we're going to end up with, again, if we look at this little cartoon, an embryo connected to the placenta via the connecting stalk. The connecting stalk not only containing the umbilical vessels, not pictured here, but the atlantois and the vitelline duct, which connects the midgut to the yolk sac. In addition, the embryo is surrounded by an amniotic cavity and lies within a chorionic cavity. And that part of the chorion that then comes in contact with the placenta is called the chorionic plate. That's where the extraembryonic somatic mesoderm is located, and during further development the amniotic cavity will be plastered up against this chorionic plate, so that we end up with a foregut in yellow, a midgut in green and blue, and a hindgut in purple, which will enlarge into a cloaca. Just looking at this in another view, head here, cephalic, tail here, the caudal area, showing you the formation of the gut. And now we can mention that the gut is going to form epithelial parts of the pharynx, form some glands like the thyroid gland and the parathyroids, give rise to the tonsils and tympanic cavity. The respiratory portion of the endoderm is going to give rise to the epithelia of the respiratory tree. And the GI part of the endoderm is going to give rise to the epithelium of the GI tract, including the glands like the liver and the pancreas, and also to the urinary bladder. Finally, we want to consider paraaxial, intermediate, and lateral plate mesoderm. Here we can see the paraaxial mesoderm, intermediate mesoderm, lateral plate mesoderm, and we will talk a little bit about their fates. First of all, if we look back on the top of this embryo, here we can see the primitive node and primitive streak. There's the notochord in the midline. There's the overlying neural plate. The neural plate is growing towards the oral plate, which we see here. And in a region in front of this, mesodermal cells have migrated from the primitive streak into a region that's called precephalic. And these mesodermal cells are going to form blood islands. The blood islands are groups of hemangioblasts, and these cells are going to give rise to some of the blood cells, as well as endothelial cells, which will go on to form some of the blood vessels in the heart. Here we can see a horseshoe-shaped heart tube that begins to form on either side of the neural tube. The heart tube begins in this precephalic area and then is going to migrate as the head fold develops into a more caudal region. This is one of the three regions where blood islands are going to form and start forming blood vessels. The three regions include the precephalic horseshoe-shaped region of the heart tube. Also, blood vessels are going to form along the yolk sac, and finally along the chorion and the connecting stalk to form the umbilical vessels. If we look at a region in this plane of section, here we're looking sagittally at the embryo, And we're going to make this cut right here, this cross section, so that we could look at lateral body folding now in the region where the heart tube is. We can see that there are two heart tubes. These are the two ends of the heart tubes. And what they're going to do is they're going to move towards each other as lateral body folding proceeds. And these two ends of the heart tube are going to move towards the midline, and they're going to fuse. So by day 21 of lateral body folding, we're going to have the fusion then of these two heart tubes, and they will be surrounded by mesoderm, splanchnic mesoderm, which will give rise to the myocardial mantle, and we will see the beginning of the formation of the heart. This is called the single heart tube stage. Once those two heart tube ends of the two heart tube meet in the midline. So now we have a heart tube surrounded by a myocardial mantle lying in front of or anterior to the foregut and suspended by a dorsal mesentery, suspended into the body cavity. And that's what we see here, the foregut, the heart tube, and the body cavity. If we look at this stage again using the University of New South Wales embryology page, what we can see is the following. We would see an embryo that looks like this. A stage 11 embryo, and then we would see a stage 12 embryo here, where you can start to see the formation of the heart. So here we can see a number of structures forming, some of the head and neck structures, the heart structures, etc. As now, by day 26 to 30, let's say an average of day 28, we begin to see an embryo that has established the vertebrate body plan. If we talk about the mesoderm, then there's paraaxial mesoderm, intermediate mesoderm, and lateral plate mesoderm. The paraaxial mesoderm is going to be split into three regions. The intermediate mesoderm and lateral plate mesoderm are also present. The lateral plate is going to be, mesoderm is going to be subdivided into two regions, somatic and splanchnic mesoderm. So as we get this lateral body folding, you can now see these layers of mesoderm. Here's the somatic mesoderm, and the splanchnic mesoderm in this region is going to surround the heart and the foregut. And then we're going to have in this region here, again it's in the region of the thorax, some of that paraaxial mesoderm, and we can see that that's subdivided into, at this point, two areas, a sclerotome and a dermomyotome. And we will talk about how this paraaxial mesoderm then is going to form a sclerotome and a dermomyotome. The dermomyotome is going to form a dermatome and a myotome, and the myotome is going to give rise to skeletal myoblasts. The dermatome is going to give rise to connective tissue, and the sclerotome here is going to give rise to the vertebra. And under the influences of some of the some of the molecules that we've talked about earlier, and which we'll get back to later on this course, we're going to see there are influences of both the neural tube and the notochord on the sclerotome and on the dermomyotome that will help direct these masses of cells to become either skeletal myoblasts in terms of the myotome, or cartilage and bone tissue in terms of the sclerotome. And so when we talk about the mesoderm, the paraaxial mesoderm is going to give rise to skeletal muscle, the skeleton, except in the region of the skull, and the dermis of the skin and connective tissue. The lateral mesoderm is going to be connective tissue of the viscera of the limbs, some of the pleura, pericardium, and peritoneum, those serous membranes, blood and lymphatic cells, as well as the cardiovascular and lymphatic systems. We've not talked much about intermediate mesoderm. This develops later and will give rise to the components of the urogenital system, including the gonads and the ducts of that